Introduction of vectors into hosts by transformation. This step is a very important step in the entire gene cloning process because it is from this point on that the multiplication of the recombinant DNA increases by several folds. So let us look at the learning outcomes of this session. Several bacteria are able to take up exogenous DNA from its surrounding, with some bacteria being more competent to take up the DNA than others. This phenomenon is what is termed as transformation. The bacterial cells are treated to make them competent cells, which means that if they are naturally not very competent to take up DNA, then they can be by being treated either with certain physical or chemical methods be made more competent, and once they are more competent, then they show a higher efficiency of transformation. There are several physical and chemical methods to carry out transformation, and non-bacterial cells too can be transfected by several of the physical and chemical methods that are used to transfect bacteria. So the question that first comes up is, why does gene cloning need transformation? Now, in gene cloning, we know that a vector, which could be either a plasmid DNA or it could be a phage DNA, is cut by a restriction endonuclease so that a gene of interest can be fit into it. Now, interestingly, both the plasmid vector as well as the phage vector and the gene of interest are either cut by the same restriction endonuclease or they are cut by restriction endonucleases in such a way that the sticky ends that are present in the gene of interest are the same as the sticky ends present in the plasmid DNA or phage DNA. Now, why is this important? It is important because if there are sticky ends, because of complementarity, the gene of interest or the DNA can anneal to the plasmid DNA or to the vector DNA easily. Once the annealing is done, then the DNA ligase ligates it and you have finally the recombinant DNA molecule formed. At this point, it is also interesting to note that cutting and ligating is not 100% process, and so there can be sometimes possible that the vector is not inserted with the gene of interest, but it can have an insert which is a DNA molecule but does not contain the gene that we are interested in. That is one point. The other point is that sometimes the vector can ligate back again without the gene getting inserted. In such a situation, it is called as self-ligated vectors. So, therefore, once you have the cutting and ligating happening, it is giving rise to a recombinant DNA molecule, but this recombinant DNA molecule may or may not have the gene of interest. Now, when you get the recombinant DNA molecule, this recombinant DNA molecule now has to be transferred into a cell. If it is bacteria or non-bacterial cell, both the cell types will have a genome of its own. Other than that, this bacterial cell will take up the recombinant DNA molecule. So this recombinant DNA molecule, which is present around in the surrounding environment of the bacteria, the bacteria can take up the recombinant DNA molecule. So this phenomenon of the bacterial DNA or any cell for that matter having the ability to take up exogenous DNA is what is termed as transformation. Now, we all know that the vectors, whether it is the phage DNA or it is the plasmid DNA, are autonomously replicating systems, and so they would multiply into more copy numbers, which means that now within the host you have several copies of the plasmid DNA. or the vector DNA. But when this bacteria undergoes further cell division, the plasmid DNA are also distributed into the respective bacteria, daughter bacteria, and within the daughter bacteria again the copy number can increase. So as you can see over here, where you have say 4 copy numbers, now when you consider in this generation 2 bacteria, you have 8 copies of the same plasmid. If this goes to the next generation, where you have 4 bacteria, you would have within each again 4 copies each. So you can see that the number of the uh, clone or the number of the recombinant DNA molecule is gradually going to increase as the bacteria keeps dividing again and again. So therefore, this bacterial DNA over here is the host organism. If it is a plant cell, then that too can be a host organism. If it is an animal cell, that too can be a host organism. Yeast can be a host organism. So there are various types of cells that can take up the recombinant DNA molecule, that is the vector with the gene of interest, and get transformed or get transfected. So effectively at this point, what we understand therefore is that if you have a bacterial cell which contains several copies of the plasmid DNA or the vector DNA, then you will get DNA in nanogram quantities. So the clone or the recombinant DNA that you will get would be in nanogram quantities. But if you take this bacterial cell and plate it on um, a nutrient agar media, then one single bacteria will divide several times to give rise to a single colony. So this single colony will have several thousands of bacterial cells that each one would have multiple copies of the plasmid DNA or the vector DNA, and therefore from a single copy colony, when you isolate or purify the plasmid DNA, you will get it in micrograms. Now, suppose you pick up a single colony and inoculate it into a liquid culture, and the liquid culture amount can vary depending on requirement. So, one can see that overnight, as the bacteria multiplies several folds, you will get millions of bacterial cells within this liquid culture, and each bacteria may contain several copies, and therefore you can understand the extent to which the clones or the extent to which there is an amplification of the clones. And therefore, in such a case where you purify or you isolate the plasmid DNA or the vector DNA, you would get the DNA in milligram quantity. So you can see how for gene cloning, which is understandably amplification of a gene of interest, you can see from one bacteria to several millions of bacteria how you are having the vector that is present inside the host getting multiplied to give rise to milligram quantities. So that is extremely important as a step in the gene cloning mechanism. And of course, this mechanism or phenomenon of a bacterial cell taking up DNA exogenously from its environment is called as horizontal gene transfer. This, when one bacteria divides into two and you have the plasmids dividing into distributed into the two, then that becomes vertical gene transfer. But when you have, say, DNA from the surrounding entering the bacterial cell or entering the non-bacterial cell, then that is what is called as horizontal gene transfer. Now, the first time that bacterial transformation was discovered or was understood was the S.F. Griffiths experiment of Diplococcus pneumonia. So when he was studying, he found that uh, during the experimentation, he found that certain live R strains of pneumococcal bacteria were transformed into live S type. So with that, he concluded that there is some transforming principle. And over a period of time, what has been understood that it is DNA that has transferred from one cell to the other cell, making the cell transformed or called as a transformant. So therefore, With SF Griffith's experiment, what was clear is that bacteria can undergo transformation naturally on its own. And it has been found that several species of bacteria are able to carry out natural transformation. For example, several bacilli species as well as streptococcal species are able to carry out natural transformation. Now, why are they able to carry out natural transformation? Because the bacteria can exist in a physiological state of competence. What does this mean? It means that the bacteria that have genes that enable DNA uptake and processing are more competent to take up DNA from outside, are more competent to uptake DNA from outside or the surrounding environment. So, therefore, this set of genes is giving rise to certain factors that are called as competence factors, and these competence factors enable natural transformation. Now, on transformation, what is possible is to get what is called as transient transformation or stable transformation. What is the difference between the two? The difference between the two is simple. In transient transformation, once the DNA is introduced into the host organism, the DNA remains in the host organisms for a small period of time or a transient period of time. It does not remain with the host until the host is alive. While in case of stable transformation, once the DNA has been introduced into the host, it will remain in the host cell until the host cell is alive. Therefore, that is the difference between stable transformation and transient transformation. Now, what can happen in transient transformation? Why is it that the DNA that has been taken up by the host does not stay for as long as the host is alive? So, what has been observed is many of the DNA, once taken up, can be degraded. That is one point. If it is degraded, then it leads to transient transformation. But if it is present inside the cell and it multiplies, then that is what is called as a stable transformation. So, say for example, a plasmid DNA is taken up and the plasmid DNA has a means of replicating, then it is able to consolidate its position or it's able to consolidate its place within the host cell. And so that becomes what is called a stable transformation. In natural transformation, what has been observed is there are several proteins that are involved in the process of pulling the DNA into the uh, wall or inside the cell through a pull and a push mechanism. So the pulling of the double-stranded DNA into the bacteria is through the outer membrane. Say for example, you're considering it's a gram-negative bacteria outer envelope. So you know that you have the outer membrane. The outer membrane has aquaporins and had certain protein structures called pill A, which is nothing but a secretin, and this is able to, through motor activity, pull or retract inside, and because it retracts inside, the double-stranded DNA that binds to the pill A is dragged inside or pulled inside. Once the double-stranded DNA is present within the outer membrane and the inner membrane, which is the periplasmic space, in the periplasmic space, the double-stranded DNA is bound to by competent factor EA, and this competent factor EA is able to associate the double-stranded DNA with competent factor EC. So effectively, competent factor EA transports the double-stranded DNA from the outer membrane to the inner membrane. Once it is present with the competent factor EC, the competent factor EC is able to translocate only a single strand from the double strand DNA. So therefore it is going to translocate, or it is going to push in a single stranded DNA, and the other strand is degraded as the COM-EC has a nuclease activity too. Now this translocation process needs energy, and that energy is provided by another competent factor, which is competent factor Fa. And this acts as an ATPase, so it is able to hydrolyze ATP to generate energy to allow translocation of the single-stranded DNA. Once the single-stranded DNA is within the host cell, if it is not bound to by proteins, then it is subject to degradation by nucleases present within the host cell. But if it is immediately bound to by proteins like DPRA or REC-A, then what can happen is that these proteins help the single-stranded DNA get incorporated into the host genome. Because it gets incorporated into the host genome, it becomes a stable transformation. So in such a case, when the host genome multiplies, obviously you have the exogenous DNA also going to get multiplied. But understandably, not all species have this natural competence. So in order to have any bacteria take up DNA in vitro, what can be done is that the bacterial cells can be made competent by some treatment. So the most common treatment that is done is that normal bacteria is subjected to treatment by cold calcium or chilled calcium chloride. The exact role or mechanism of calcium chloride in the ability to make the bacterial cell more competent is not very clear. But the hypothesis is that because it has a positive charge and it can bind to the negatively charged cell wall of the competence of the bacterial cell, it acts as a bridge between the negatively charged vector DNA and the wall of the bacteria. So effectively, anchorage of the plasmid bound anchorage of the vector DNA to the surface of the bacteria is thanks to calcium chloride. However, calcium chloride is not able to transfer or translocate the plasmid DNA within the host cell. That is done by a process that is called as heat shock. So what is done is the bacteria attached to the plasmid DNA is subjected to 42 degrees centigrade for 2 minutes. Now this is important and this has been standardized. If you leave bacteria in 42 degrees centigrade for a long period of time, then the bacterial cell will die because of stress. So it has to be kept only for so much so of time that does not affect or harm the competent bacterial cell. Now, what is the purpose of heat shock? So, what has been again the exact mode of action or the exact way by which the heat shock works is not very clear. But what is understood is that when you have the DNA already bound to the cell wall of the bacteria, and if you subject it to heat shock, then it may be possible that the wall forms pores. And through those pores, the plasmid will get transported into the cell. So it is a very simple concept, and this simple concept actually is responsible for increasing the transformation efficiency. So a normal bacteria is made competent by by the chilled calcium chloride. So it is soaked in chilled calcium chloride for a stipulated period of time. And then these competent cells are subjected to or are exposed to the DNA to which it binds and then is given heat shock. So, for example, in the labs generally, when one has to carry out transformation, one of the bacteria that is commonly used is E. coli DH5 alpha. Please pardon, this C should be a small c. Now, E. coli DH5 alpha is actually an engineered bacteria. This engineered bacteria is more competent. So, the engineering has been done to make this E. coli more competent. So there are three mutations that are carried out in E. coli to lead to formation of E. coli DH5 alpha. What are the three mutations? One mutation is that it, it is mutating a gene that enables homologous recombination, and so when that gene is mutated, then one doesn't observe a homologous recombination. So what is the implication of this? The implication of this is that the E. coli DH5 alpha, which takes up the exogenous DNA, the exogenous DNA will be present in the host, but it will not get integrated into the host genome. The second mutation is that the mutation is done in an enzyme that acts as an endonuclease. If that endonuclease is inactive, then the DNA that has been taken up by the E. coli DH5 alpha will not be degraded. So these two are very advantageous for E. coli DH5 alpha to not just be competent but also to protect the DNA that has been taken up. The third is that a gene has been mutated that enables selection of a transformant. We understand what transformant is. If the vector DNA does not have the gene of your interest, then it is a non-transformant. But if the vector DNA has the gene of your interest, then it is transformant. But transformant here is talking about the host cell. If the host cell takes up the recombinant DNA, then it is a transformant. And if the host cell does not take up a recombinant DNA, then it is a non-transformant. So it is important to distinguish between those E. coli that have been transformed and those E. coli that have not been transformed. And a gene that is mutated enables the selection of transformant from a non-transformant. So effectively, this method of transformation in vitro is a chemical method of transformation. And this method has been modified in several ways. So, for example, one has observed that calcium chloride is used along with PEG, that is polyethylene glycol. And polyethylene glycol also is enhancing the attachment of the DNA to the wall of the bacteria. So, it is helping in this first step. So, it is making the cell competent by ensuring that the DNA is at least bound to the surface of the bacteria. What is also used is nanoparticles. So, like for example, silver nanoparticle, copper nanoparticle, these are also used. So, because they are very small in size, and if they are bound to the bacteria, they are bound to the vector DNA, then along with the nanoparticle, you can have the vector DNA also entering the cell. So therefore, this method of transformation is the most commonly used chemical method in vitro. Now we go to physical methods that are used to transform bacterial cells. But here you must understand that it is not just bacterial cells that can be transformed through physical methods, but also non-bacterial cells like plant cells, animal cells, fungi, insect cells, etc. All of these can be transformed by the physical methods. The most common physical method that is used is DNA electroporation, and the concept is very simple. So suppose you have a cell surrounded by exogenous DNA in a solution, and what is introduced is a set of electrons, positive and negative, and an electric current is passed. So therefore what is generated in the solution is an electric field. And based on that electric field, what is observed is that pulses of the electric field can kind of destabilize the bilipid layer. Now, these pulses, therefore, when they destabilize the bilipid layer becomes more porous. And through the pores that are formed, the DNA, which is present in the surrounding, can enter the host cell. When the pulse is removed, then the bilipid layer again reassembles or rearranges themselves to become less porous or not porous at all. So what has happened in the process is DNA from outside is able to get into the cell thanks to the formation of the pores, and these pores are formed thanks to the electric field that has been created. That is the reason why it is called as electroporation, that is formation of pores using electric field. So as you can see over here too, one thing has to be kept in mind, that if the cell is exposed to electric stimulus for a long period of time, then the cell can get destroyed. So therefore, it is important uh, to kind of standardize how much amount of pulse for how much time has to be given or has to be subjected to the cells that have to take up the DNA. So as you can see over here, when it is less than threshold voltage, it is the cells are intact. Okay, you increase the voltage, and you can see that the poration increases. And when you decrease the voltage again, you will find that the lipid bilayer reseals again. So this is a very important principle based on which you can have uptake of DNA by cells. On similar lines is what is called as magnetoporation. In magnetoporation, the DNA is bound to a magnetic bead, and now a magnetic field is created. So, as the magnetic field is formed, the beads are pulled towards the magnet, and in the process, as it pulls towards the magnet, it would actually attach to the surface of the bacterial cells. And thereby, what happens is, because there's a pull by the magnetic field, it can also enter within the host cells. So therefore, this is another concept by way of which you can have transformation happening. Then you have what is called as sonoporation. Sonoporation is using sonic waves. So sonic waves are also able to create pores on the wall of the bacterial cell or the non-bacterial cell, and the DNA can enter the host cell through those pores that have been formed. Similarly is optoporation. In optoporation, what is used is a laser beam, and the laser beam is able to again create pores on the bacterial cell or the non-bacterial cell. An exogenous nucleic acid can enter the cell through the pores that have been created because of the treatment or subjection to the laser beam. In all these cases, of course, what is important is that how much or what is the intensity of the sound wave, or what is the intensity of the laser beam, for how long the sound waves have to be present, have to be given, for how long the laser beam has to be there to form pores. All that has to be standardized. Now, transformation in vitro based on another principle is also possible, and the examples of that is particle bombardment method, also called as the bioelastic method, and DNA microinjection. So, in the particle bombardment method, which is also called as particle gun method, you can see that here this is a simulated firing piston. So this acts as a gun, and this over here is the bullet, or what is called as a macro projectile. And at the end of the macro projectile, you have what is called as micro particles. So these micro particles can be gold nanoparticles, and these gold nanoparticles are coated with the DNA of interest. So once you have this coated with the DNA of interest, and the piston or the gun is put in the chamber, which has a certain medium that will allow fast traveling of the microparticles, you can see that on the other end the target tissue has been immobilized, so that when the gun particle is shot, the microparticles will move from this thin uh, uh, pinhole or thin um, area to reach the target tissue. And therefore, it basically means that the microparticle pierces inside the host cell. And that is how you have the DNA from outside getting introduced into the living cells. Another example is that of DNA microinjection. This too is a method that is commonly used. The concept is simple, but the instrumentation is quite expensive, because one has to use very precise uh, um, and special uh, instruments, conditions. So the idea generally is that the cell that has to be injected with the DNA of interest is generally through suction immobilized. And this immobilized host cell, when observed under the microscope, a thin glass needle attached to a syringe pierces, so the needle pierces into the nucleus of the immobilized host cell, and within the syringe is present the DNA of interest. So when one injects it, what happens is the DNA directly enters into the host cell because of the piercing of the needle through the cell wall. And this piercing is so fast, or this piercing is so thin, that the cell wall and the cell membrane is never going to get affected or never going to get disrupted. And so this is a very good method again of transformation in vitro. So let us look at the conclusions. Natural transformation by bacteria has been observed as they have a set of genes that can uptake DNA as well as process the DNA. The most common chemical method is using chilled calcium chloride to make the bacteria competent, and once the bacteria is competent, then give a heat shock to ensure that pores are formed, and through the pores the DNA is taken up. Several physical methods like electroporation, magnetoporation, sonoporation, optoporation are using the principle of opening pores in the cell wall and membrane. And with the opening of the pores, again, therefore, you can have DNA from outside entering the cell. DNA microinjection and ballistic particle bombardment method are based on piercing through the outer envelope to introduce the DNA. Thus, the phenomenon of transformation enables horizontal gene transfer in bacteria as well as non-bacterial cells. This step is important in gene cloning, but it has a challenge, and the challenge is that of selecting a host cell that has the recombinant DNA, and such host cells are called as transformants. So it is important to select those host cells that have taken up the DNA. Thank you.